Hello, my name is Palahs Gupta, and today I'd like to discuss my research on the advances in viral vector technologies in regards to delivering gene therapy. What even is a viral vector? A viral vector is a method to deliver genetic material into the cell. Those little spikes on the sides of a virus are actually proteins that can unlock a cell, which is why viruses are so good at getting inside of the cell. The question lies, how does this relate to gene therapy at all? When trying to get a gene inside of a cell, you can put it inside of a virus, and then the virus can be sort of the taxi to get it inside of the cell because it's really good at that. That's sort of its job. So if you can just put genetic information inside of a virus, how do you choose which virus you want to put it inside of? Well, there are three main ones at play: adenovirus, adeno-associated virus, AAV, and lentivirus. Each of them have their pros and cons. Adenovirus, on one hand, is super big, which just means it's really good for large-scale projects like cancer therapy, and it was even used in the Johnson & Johnson COVID vaccine. On the other hand, it carries a crazy immune response, which means people will really have a struggle fighting off with it, and it'll have immense side effects. AAV, on the other hand, though still having considerable immune response, is less so. But on the other hand, it's much smaller, which means it can carry significantly less genetic information. Lentivirus sort of takes its own spin on things. Instead of being used inside of the body in vivo, it's used in vitro, which means you can put it inside of a petri dish, and it has a property which means it's integrative. And what that essentially does for the virus, it can be put inside of a petri dish and it can spread to a bunch of cells inside of a colony. And if those cells can be put inside of the body, you don't really want to use the lentivirus inside of the body because since it can spread, it can integrate, you don't really want it spreading to places it can't, especially because it is permanent. There are flaws with viral vectors though, but there have been solutions as well. Chimeric viral vectors are one incredible solution in regards to antibodies. This example over here shows 85, which is incredible at targeting the liver, and 8035, which is less so. But 85 has one major flaw in the fact that humans have already developed antibodies, which means it's completely neutralized because it's easily detected. 8035, on the other hand, though not really effective inside of the body, cannot be detected because it's not the most impactful. So if you keep the protein of 8035 to trick the receptors that try to block 85, all of a sudden you have something really good at targeting the liver while also being really undetectable. Another massive issue is that viral vectors, when they're inside of the body, don't really have a spatio-temporal awareness. But the TET-on inducible system is one way that they've tried to combat that. The TET-on system is a way to control when the gene is turned on. It uses a modified transcription factor that only activates when the antibiotic DOX is present. Without DOX, the gene does nothing. But with DOX, it binds to like a protein, which then attaches a DNA-specific sequence called TRE, which basically just activates the gene. And you can see the gene is activated here. Overwhelmingly, viral vectors have improved so much in the 21st century, and there's only more innovation to come. Thank you.